Digestion means breaking of complex food to simpler substances which could be easily absorbed from the body. Now, proteins, as we know, are the building blocks of our body, which helps our body to grow. Digestion of proteins is the conversion of proteins into such substances which could be easily absorbed from our intestines. Now, those substances are amino acids. Amino acids are the smallest monomers of proteins which could be easily absorbed from the intestine. Digestion of proteins usually happen in three main sites. First is the stomach, which is the major organ for protein digestion. Second is the duodenum. Duodenum is the first part of the small intestine which possesses two other parts, namely jejunum and ileum. And the third site where digestion of proteins completes is the rest of small intestine, which includes the other two parts that are jejunum and ileum. Let us begin with the digestion of proteins. The first site which we discussed was the stomach. Stomach is an inverted J-shaped structure located inside the abdominal cavity. Now, when the bolus of food is the chewed food mixed with saliva which passes down the esophagus into the stomach. Now, when the bolus of food enters the stomach, it stimulates the G cells, and when G cells get stimulated, they release a hormone which is known as gastrin. Now this gastrin is released into the blood, which again re-stimulates gastric glands. Gastric glands possess receptors for these hormone gastrin, and when gastric glands are stimulated, they stimulate the further cells located inside them. The cells which are stimulated are oxyntic or parietal cells. Second, chief or peptic cells, and some other cells. When oxyntic and parietal cells are stimulated, hydrochloric acid is released, thereby making the medium inside the stomach acidic of a pH of about 1.3 to 1.8. That is highly acidic. When chief cell or peptic cell are stimulated, they release Pepsinogen. Now, this is an enzyme we will talk about later. And other cells of the gastric glands, they release prorenin. What this Pepsinogen, HCl and prorenin do, we'll have a look at it. First of all, we need to remember that wherever the suffix ogen, like here, wherever the suffix ogen is present, this means that the enzyme is inactive and needs to be activated in order to perform its physiological function. And wherever a prefix pro is present, that again means that the enzyme is inactive and needs to be converted into its active form in order to perform its normal physiological function. So, whenever bolus of food enters, HCl is released in large amount. That means whenever we ingest something, the medium inside our stomach becomes highly acidic, and with the release of pepsinogen and prorenin. Now this pepsinogen is inactive and needs to be converted into its active form, which is pepsin. and prorenin needs to be converted into its active form, which is renin. And both of these are activated with the help of hydrochloric acid. This acidic medium provided by hydrochloric acid is important for the conversion of pepsinogen and prorenin into their active forms, that is pepsin and renin respectively. When proteins are present in the bolus of food, they are acted upon by the activated pepsinogen and prorenin, that is pepsin and renin, to form polypeptides, peptones, and proteoses. Now, these are the comparatively simpler form of proteins, which are formed with the help of enzyme pepsin and renin. Now, how renin is specifically important is milk, which we ingest, contains a protein known as casein protein, which cannot be broken down efficiently by pepsin. Therefore, it requires a special enzyme, which is renin, which converts casein into calcium paracasinate. This calcium paracasinate is easily now converted by pepsin into polypeptides. Now, the action performed by renin on casein protein is known as curdling, as it curdles this protein. The action performed is known as curdling of milk. Now, this is important, as for us to utilize the protein of milk, that is casein, efficiently, renin is very important. But this renin, or rather prorenin enzyme, is released only in children. Therefore, milk is considered as an important ingredient in their diet. So, the last products which are formed in the stomach after the partial digestion of proteins are polypeptides, peptones, and proteoses. Now, the partially digested proteins which we were having were polypeptides, peptones, and proteoses. Now, these passes down from stomach into the duodenum, which is our second site for digestion of proteins. Duodenum is the first part of small intestine and a very important part of human anatomy, which plays an important role in the digestion of food. Now, this part is known as the duodenum. The bolus of food passes from stomach into the duodenum. Now, inside the duodenum, there opens the duct of pancreatic gland. Pancreas is a heterocrine gland located just below the stomach. Pancreas, being a heterocrine gland, has two functions. One is the endocrine function, which is concerned with the release of hormones, and the second is exocrine functions, which is concerned with the release of enzymes, particularly digestive enzymes. We are not concerned here with the endocrine function of the pancreas, as it deals with the hormones, and we'll see that in later lectures. Particularly, we are concerned about the exocrine functions, which deals with the release of enzymes. Now, pancreas basically is known as the complete digestive gland because it releases digestive enzymes for digesting all the components of food, that is carbohydrates, fats, and proteins. All the three components are digested with the help of the enzymes released from the pancreas. Enzymes which are released from the pancreas, which are responsible for the digestion of the semi-digested proteins, polypeptides, peptones and proteoses. They are, first is trypsinogen, second chymotrypsinogen, third carboxypeptidase, and last elastase. Besides these, nucleases is also released, but these four are major in digesting these incompletely digested proteins. Now, as we remember again, the ogen suffix, whenever added, renders inactive form to the enzyme. So trypsinogen and chymotrypsinogen both are inactive enzymes and need to be activated before they can act upon the proteins. So the activation of pancreatic protein enzymes, there runs a cascade mechanism, that is, activation of one enzyme leads to the activation of another enzyme, or in other words, the activated product of one enzyme leads to the activation of the another enzyme. Let us understand. From the small intestine, or rather the duodenum, there is release of enterokinase. Now, enterokinase is a non-digestive enzyme which is released from the duodenum itself. Literally, if we break the word, entero means intestine and kinase means motility. It basically increases the motility of the intestine. It is a non-digestive enzyme but plays a very important role in the activation of trypsinogen. Trypsinogen is activated into trypsin, its activated form, with the help of enterokinase enzyme. Now, as we considered earlier that the pancreatic enzymes are activated with the help of a cascade mechanism, this activated trypsin leads to the further activation of inactivated chymotrypsinogen into chymotrypsin. So it is trypsin which helps chymotrypsinogen to get activated into chymotrypsin. Now, these both activated trypsin and chymotrypsin, along with carboxypeptidases and elastases, are responsible for the conversion of these polypeptides, peptones and proteoses into dipeptides and tripeptides. Now, these dipeptides and tripeptides pass down from the duodenum into the other parts of small intestine, that is jejunum and ileum, where they are acted upon by the intestinal juice. Now, these intestinal juice also possesses many enzymes besides the enzymes required for converting proteins into their simpler form. Intestinal juice possesses eripsin, which is a collection of enzymes responsible for converting dipeptides and tripeptides into their finally digested or the simplest form, amino acids. Now, Eripsin is a group of basically two enzymes. One is the dipeptidases for converting the dipeptides or digesting the dipeptides, and second, tripeptidases for digesting the tripeptides. Now the amino acids so formed are the simplest form of the proteins which could be easily absorbed by the intestinal gut into the bloodstream. Now this absorption could be both active and passive, that is, may or may not require energy consumption for the absorption of amino acids. That's all about digestion of proteins. Thanks for watching.